because this is not the first time we hold a seminar like that, and so this is an online journal club that we use to discuss recently published papers, and the topics are mainly focused on biomedicine and the nanoscience. And in these seminars, researchers have an opportunity to communicate science to a broader audience, while the audience can get answers to their most tricky questions from the experts who publish those papers. So the seminar is going to be recorded. It's a 45 minutes presentation, and then 45 minutes maximum for Q&A. And so today we are honored to host Professor Dr. Stephen Goodman, presenting a study of the role of extracellular DNA structure in the offenses and defenses of the host and biofilms. First, it's a presentation, and then there will be Q&A session, but the questions can be posted in chat during presentation. I would like to give a short introduction to Stephen Goodman's biography. He's a PhD and a professor of pediatrics at the Ohio State College of Medicine, director of the Oral Microbiology Research Affinity Group, and the principal investigator in the Center for Microbial Pathogenesis in the Abigail Wexner Research Institute of Nationwide Children's Hospital. Stephen has spent 20 years on the faculty at the Ostro School of Dentistry of the University of Southern California, where he was the chair of the Division of Biomedical Sciences and director of the university-wide Microbial Systems Institute. He has published 79 peer-reviewed manuscripts and recognized expert on the formation, structure, and function of exopolymeric substances and gene regulation in bacterial biofilms. Stephen is a biochemist by training, receiving his doctoral degree from the Bloomberg School of Public Health at the Johns Hopkins University. So with that, I would like to welcome you one more time and pass to you the screen. So the word is yours now.
Thank you for the invitation and for allowing me to present our view of bacterial biofilms and how eDNA structure plays a role. Can you all see the slide? Mm-hmm. Yes, okay, good. All right, so I am going to present the paper, but I want to give some background, and the background is actually going to take about a third of the talk so that everybody is up to speed and understands the history and also the biological implications of what we've discovered. So just to refresh everybody's memory, bacterial biofilms are communities of bacteria. They can be single species, they can be multi-species, but more often than not, they are multi-species. These bacterial communities can either be in an aggregate form or they can attach and then form aggregates. And so that's a whole discipline to distinguish between aggregates and bacteria that have attached. And so at biofilm meetings, people have very spirited discussions about whether they're different or not. Having said that, the natural process for an adhering biofilm is for planktonic or free-living bacteria to bind to a surface and then to proliferate. And during that proliferation, there are lots of physiologic changes, including cell-cell communication, transport, metabolism, but probably the most important hallmark of biofilm formation is the creation of an extracellular matrix. And that matrix pretty much consists of every biomolecule that's inside the cell is outside the cell. And so every bacterium has a different program of materials that they will release. And so the extracellular polymeric substances, which are the matrix, otherwise called the EPS, can consist of multiple different matrix materials. And this complicated what kinds of structures that might form between multi-species communities. The problem is that when you have multiple species with multiple matrix materials, how do bacteria interact productively if the matrix are different? And so this has been one of the big issues in biofilm study. So despite the fact that there are all of these different types of biomolecules that are present in different species, the EPS typically, and I will say universally, contains extracellular DNA. The source of the DNA is at least from the bacterium themselves, and we won't really talk about it, and I'm certainly happy to answer questions in this regard. The bacteria can also usurp DNA from other sources, and we'll talk about that later. So back in 2008, I was in the audience attending a seminar given by Lauren Bakelitz, who is my primary collaborator, and she showed this image. The green dots are the bacterium non-typal Haemophilus influenza, which is the etiologic agent of otitis media, middle ear infections. But in the foreground, she was baffled by this blue lattice, which is DNA. And she told the audience to ignore the DNA because that wasn't really what she was looking at. She was looking at the bacterium because she studies otitis media, and she was trying to create a vaccine that was specific for non-typal Haemophilus influenza. However, when I saw those structures, it reminded me of certain kinds of intracellular DNA intermediates, replication forks, or Holliday Junctions, which are recombination intermediates. Now, we had discovered surreptitiously that back in 1994, that a protein that is ubiquitous throughout all eubacteria, called the DNABII proteins, which has two members, HU and IHF, that this protein was found extracellularly. And when we started to look at this, we found that other people had discovered the same thing in various species, that the DNABII family was typically released in large quantities, not necessarily by autolysis or any other kind of lysis, but through some kind of program release. And these proteins bind with very high affinity to Holliday junctions and replication forks. Why do they form so well? Because when this family of proteins binds to DNA, it bends DNA. And if the DNA is already in a bent conformation, then these proteins bind with even higher affinity, typically 1,000-fold tighter to bent DNA than to non-bent DNA. Now, these proteins are incredibly important intracellularly. I'm not going to go through all the functions that they have, but they share a common secondary structure and mechanism for binding DNA, which includes two antiparallel beta ribbons that bind DNA 9 base pairs apart and causes two dramatic kinks in the DNA that causes the DNA to bend almost in a perfect U-turn. And so after Dr. Bakelitz gave her talk, I approached her and I said, I know why your structure looks that way. We know about this protein that binds to Holliday junctions because they look like bent DNA. And I predict that if you were to do immunofluorescence of your DNA, that at every single vertex, in other words, every place where the DNA crossed, that's where you would find the DNABII proteins. And you wouldn't find them anywhere else, because where the DNA strands cross, the binding affinity should be 1,000-fold higher than any of the other strands. And indeed, that's what she found. However, the question then arose, what was the protein actually doing at those vertices? And so we argued that the matrix was actually held together by these proteins. And if you could remove those proteins, then the eDNA structure would collapse. And as a consequence, the biofilms would be disrupted. And so we addressed this by doing a static biofilm assay. This is a confocal image of on the left is an NTHI biofilm. You can see the XY representation and the ZX representation, in other words, the height. And when we add naive serum to a biofilm, you see that there is still a very strong biofilm present. The biofilms have been stained with a live dead stain. Green is alive, red is dead. You'll see that both images on the left and right panels, there's no red, so no one is dying. But when we add antibody that is specific to the DNABII proteins, even though the biofilm is 24 hours old, the antibody is able to disrupt the biofilm. Now, one thing I haven't told you is, or two things I haven't told you is, one, once biofilms form, they are highly resistant to antibiotics, typically 1,000-fold higher minimum inhibitory concentration relative to planktonic bacteria. And second, the biofilm matrix does not readily allow antibodies to penetrate into the biofilm. And yet, this antibody was able to disrupt the biofilm. And so other people have tried to design antibodies, and they typically don't have any effect on extent biofilms. We have used this approach. This is an incomplete list of all the different types of biofilms that we've used our generic antibody on. You can see it's quite extensive, contains the usual suspects. It also contains multi-species biofilms like sputum samples from cystic fibrosis patients. And so our argument that this is a universal matrix structure is consistent with the notion that it does not matter what the origin of those bacteria are. They all, once they form a biofilm, they will collapse when we add the antibody. So what's actually going on? This is a cartoon. The biofilm bacteria are represented in green, the DNA in white, and the DNABII proteins are the orange circles. And you can see that the DNABII proteins prefer to bind to the vertices of the DNA, but some of them are in equilibrium with the bulk media. When we add antibodies, the antibodies are able to sequester the free protein. That shifts the equilibrium, eventually causing catastrophic collapse of the biofilm. Now, we've been able to demonstrate this. This was published in 2014 in a paper by Broxson et al., where we were able to immobilize antibody in a transwell device where the antibodies were in the apical chamber and the biofilm was in the basolateral chamber. And so the antibodies never touched the biofilm at all. The only thing that could pass through the septum between the apical and the basolateral chamber were small molecules like the DNABII proteins. And because the antibodies were specific for DNABII proteins, we concluded that the antibodies were simply driving the equilibrium, causing catastrophic collapse of the biofilm. So what is the nature of the eDNA lattice structure in the bacterial biofilm? And this is the prelude to the zDNA story. So as I told you, the DNABII proteins bind to DNA and bend DNA. But if the DNA is already in a bent conformation, they will bind with higher affinity. And one of the structures that the DNABII family binds to with very high affinity are Holliday Junctions. And so when I started telling this story 12 years ago, I would say this array that we see, this lattice of DNA, looks a lot like DNA origami or a series of a secondary array of Holliday Junctions. And people would sit in the audience and they would nod and say, yes, yes, of course. Well, I incorporated it into a grant and I got my hands slapped because I did not prove that they were Holliday Junctions. And so we spent several years proving that these structures were functionally similar to Holliday Junctions. So Holliday Junction resembles a cruciform. It has 4 strands and it is typically associated with homologous recombination. And so the first thing that we did, and this is work published back to almost 2.5 years ago now, is we used an antibody that was specific for Holliday Junctions and cruciforms. And we showed that when we made biofilms, that everywhere that the vertices crossed, In other words, the same places where the DNABII proteins bound, the antibody recognized those same areas, indicating that those structures were highly similar. Whether the biofilms were derived from ex vivo samples from middle ear infections or whether they were derived from cystic fibrosis sputum samples. So we decided to do an equivalency experiment to prove this. So within bacterium, and it's not a universal protein, but it is fairly ubiquitous, there's a protein called RuvA. And we're going to talk a lot more about RuvA in a few minutes. But RuvA, sole job is to bind to Holliday Junctions with very high affinity. And once it binds there, it stabilizes those Holliday Junction structures. So, and it does not bind duplex DNA. So our experiment was as follows. We would take a biofilm. We would treat the biofilm with anti-DNABII protein, in this case anti-IHF, which would normally disrupt the biofilm. But at the same time that we were adding the anti-IHF, we were going to add RuvA simultaneously. And if we were correct that the role, one of the roles of the DNABII proteins was to stabilize those Holliday junctions, then RuvA should be able to replace IHF, and i.e. it would be an equivalent binding protein. And that's exactly what we saw. We looked at 3 different species of bacteria, uropathogenic E. coli, non-typal Haemophilus influenza, and Staphylococcus epidermidis, all very strong biofilm formers. And so if you look in panel A, you see that if we add anti-IHF, the biofilm gets disrupted, but if we add anti-IHF and RuvA together, that there is no disruption. And let's just use the uropathogenic E. coli as an example. We see that the biofilm biomass is strongly reduced when we add anti-IHF. And let me also say that typically we don't add, we add just enough antibody to be able to see about a 50% reduction because this keeps us in close to the linear range of being able to tell how much biofilm is actually being disrupted. If you add more antibody, we can drive the biofilm down to a monolayer, which is exactly what's predicted, and it is dose dependent. If we add IHF, anti-IHF, and we add RuvA simultaneously, we don't see disruption. And if we use other proteins, the anti-IHF works just fine. So HNS is another nucleoid-associated protein. It does not bind to Holliday junctions and does not have an effect. When we add anti-IHF, we've done some, we see that there are rheological changes. We've measured the Young's modulus of biofilms that have formed. And basically, the Young's modulus is an indicator very qualitatively of stiffness. When we add anti-IHF, the stiffness of the biofilm goes down. When we add RuvA, there's no change. And we add anti-IHF and RuvA, there's no change. And so as a consequence, we conclude that when RuvA replaces IHF or HU, any DNABII protein, it does so and maintains the structural integrity of the biofilm, not just the biomass. So RuvA is part of a complex of 3 proteins inside the cell that's required for resolution of Holliday junctions. RuvA binds to Holliday junctions. RuvB is a helicase that binds to RuvA bound to Holliday junctions and allows branch migration. And RuvC cleaves the complex, the Holliday junction, after homologous recombination is complete. So we argued, oh, we're stuck. Try this, here we go. So we argued that once we added RuvA to the complex, that if we added RuvB and C, that rather than stabilize the complex, the biofilm, that the biofilm would be disrupted. And this would argue that not only does RuvA recognize the Holliday junctions and stabilize them, but the RuvA-B-C complex, which should cleave Holliday junctions, should disrupt the biofilm. And we showed this for the same 3 bacterium. We can simply review uropathogenic E. coli. We see that anti-IHF reduces the biofilm, disrupts the biofilm. RuvA by itself does not, RuvA-B does not, but when we add RuvA, B, and C, regardless of the bacterium, we see a reduction in the biofilm biomass, indicating disruption of the biofilm. So the conclusion is that these vertices are Holliday Junction-like. It doesn't mean they're exactly Holliday Junctions. They're just similar enough that RuvA recognizes them. And we've also looked at another Holliday Junction resolvase, RusA, R-U-S-A, and it likewise disrupts biofilms. So everything is consistent with the functional ortholog. So there is another mystery about this DNA, which has baffled biofilm researchers from the very beginning. Despite the fact that there's a lot of DNA in the biofilms, biofilms are resistant to DNase. And the question is why? So this experiment is now 20 years old. It's hard for me to believe because I remember when Cynthia published this. It was published in Science. They don't publish these anymore. They're called Brevia, or they were called Brevia. One page, one figure tells the whole story. And so this was one of those things that changed my life as well as everybody who works on biofilms. What Cynthia did was to take Pseudomonas aeruginosa, and she mixed in pancreatic bovine DNase, and she was able to show that when she added the DNase with the bacterium, that they did not form biofilms. However, as she waited and allowed the biofilm to establish, the biofilms became more and more resistant to the DNase. And so she presented this data at a meeting, ASM biofilm meeting in 2003. Everybody ran out and looked at their biofilms, and we were pretty much able to recapitulate the same phenomena. And the conclusion was extracellular DNA is important for biofilm formation, but once the biofilm is formed, it must not be important because the DNA does not get cleaved anymore. And so that might well have been true, except that Lauren Bakelitz and my work had shown that the DNA is, and I'm not going to show any of that data in the interest of time, that Lauren Bakelitz and I had shown that DNA is important throughout the course of biofilms. In fact, we've grown biofilms up to 2 weeks old, and they still are able to be disrupted by anti-DNABII, and if there's no DNA present, then there's no biofilm present. So this really forced the question, why are biofilms, when they mature, resistant to DNase? Well, I will tell you, we tried every nuclease I could possibly imagine, and I'm happy to discuss that later to tell you about what all the nucleases we tried, nothing worked once the biofilms became mature. But I had recalled when I was a postdoc, I got to know Alex Rich very well. And Alex Rich basically discovered, I mean, they're arguably discovered zDNA, but there is a form of DNA that's DNase resistant, nuclease resistant. And that is a left-handed form of DNA called zDNA. That was originally, the structure was originally solved by Alex Rich and coworkers back in 1979, although there was evidence that some weird things were going on with DNA that was in high salt. zDNA is left-handed. It is in equilibrium with bDNA. bDNA is the standard right-handed helix that we all know and love. zDNA has some very interesting properties in addition to being resistant to nucleases. In fact, no DNA binding proteins that bind bDNA bind zDNA. Not surprising, the helical structure is completely different. The Watson-Crick base pairs are for the most part conserved. So A and T bind the same way in Z and B, and G and C still form base pairs as well. The phosphate backbone of zDNA is much closer together than in bDNA, And those electrostatic repulsions tend to favor bDNA under physiologic conditions, at least some most physiologic conditions. When the DNA is supercoiled, then it reduces the energy of activation, and there's a equilibrium that tends to shift towards zDNA. And so when zDNA was discovered, the only way you could really drive B into Z is at very high salt concentrations. For example, 4 molar sodium chloride. So the first question we wanted to ask is zDNA present in vitro biofilms? And so we looked at some of the usual suspects. They're listed on the right of each panel. bDNA is in blue. This is all immunofluorescence. So, and these antibodies are highly specific. If you read our cell paper, we went to great lengths to show that the antibodies are highly specific. bDNA is blue, zDNA is yellow, and you can see, depending on the species of biofilm, there's always B and Z present, but some bacterial biofilms have more Z than B and vice versa. We then looked at ex vivo biofilms from the middle ear of chinchilla with otitis medius, in addition to CF patients. And this, we would argue, is far more realistic than the in vitro biofilms. But again, we see a lot of yellow compared to the blue. And you can see that the yellow tends to be in long strands, long persistent strands, whereas the blue is more diffuse. The next question we wanted to ask, because this is part of our model, is that as biofilms develop, there would be an accumulation of zDNA. That would have to be true, otherwise they wouldn't be DNase resistant. And although there is more zDNA that is put out into the extracellular matrix with time, there's also a lot more bDNA there too. The only exception so far, and this is the only one that we've only looked at these 3 bacterium, is non-typal Haemophilus influenza. At 1 week, there is no detectable bDNA. It's all zDNA. And I can tell you from personal experience that NTHI creates one of the stronger biofilms that we've ever looked at. Klebsiella pneumoniae and uropathogenic E. coli tend to make more flimsy biofilms. And so we concluded towards the end of our work in the cell paper that zDNA was highly important for creating rigidity in the biofilm. So the question then becomes, how important is the bDNA? So we already know if we add DNase to a mature biofilm, we don't see any changes in the structure of the biofilm or the biomass of the biofilm. But the first author on the paper, John Buzzo, did one of those Saturday afternoon experiments where he didn't tell me what he was going to do, but he decided to add DNase to a biofilm and then perform immunofluorescence. And we knew it wasn't going to do anything, but lo and behold, what he found was for 3 different distinct species of biofilm, that the bDNA was digested, but the zDNA was unaltered. And of course, the biofilm biomass did not change. And this argued that the basic biofilm structure was not contingent on bDNA at all, and it was reliant on zDNA. To confirm this, we did 2 different experiments. One, we added cerium chloride, which we showed, and others have sort of showed, that cerium chloride, which is a polycation known to drive the equilibrium from B to Z, that when we added cerium chloride at concentrations that convert B to Z, but do not interfere with the physiology of non-typal Haemophilus influenza, actually caused the biofilm to grow in size. And what does that mean? And so in all biofilms that are static biofilms, there is an equilibrium between planktonic and biofilm bacteria. The bacteria never end up completely in the biofilm, ever. And so if you create conditions that drive bacteria into the biofilm, you will see a larger biofilm because you're getting more planktonic bacteria driven into it. And so for non-typal Haemophilus influenza, typically between 30 and 40% of the bacteria in a static biofilm tend to be driven into the biofilm. When we add cerium chloride, that number goes up significantly. Now, thanks to Alex Rich, we also know that most intercalating agents, in other words, molecules that bind DNA by inserting themselves between the bases, drive bacteria into the B state and tend to disrupt Z. And so we wanted to pick a intercalating agent that was known to be non-toxic. And these experiments were done before our former president of the United States decided it was going to cure COVID. So we feel like we started the chloroquine bandwagon, but now it's somewhat tainted because of all the histrionics that went on with COVID. Nonetheless, when you add chloroquine to biofilms, it does disrupt biofilms. So the question is what is the chloroquine actually doing? You could argue it could be doing a lot of things. We're arguing it's driving the Z back into B DNA. To prove that, we showed that biofilms that normally would be recalcitrant to DNase become sensitive to DNase when we add chloroquine. Likewise, we have a sputum assay that shows that in the presence of chloroquine and DNase, we get full disruption of sputum, whereas DNase and chloroquine at lower concentrations fails to show a strong response. And so all this indicates is that by adding chloroquine, we are likely driving zDNA back into B, and as a consequence, making the biofilms less stiff. The other thing, and I'm going to go through this quickly because I want to go through other things that are very important. We argue, one of the arguments that we got from one of the reviewers, and it's a very good argument, so we incorporated it into our paper, was when you create the zDNA and you get rid of the bDNA, you can't argue it's in an equilibrium anymore. Because if the bDNA is gone, then you should be driving the zDNA back into bDNA. And so adding DNase should always disrupt the biofilm eventually, because you're driving the equilibrium. And that's not what we see. Once the zDNA forms, the zDNA is locked in. So our argument was that the zDNA would be dependent on the Holliday Junction structure, that origami array. And so we used the resolvase RuvA to show that when we added RuvA in small quantities, the zDNA went away for non-typal homophilous biofilms. And so again, this seems to be that the structure that's formed with these Holliday Junction arrays not only incorporates zDNA, but rely on these Holliday Junction-like structures. I'm going to skip this, and I'm just going to say that when you add more HU, it drives DNA. This also is consistent with this notion that the Holliday Junctions are incredibly important for the maintenance of zDNA. And then the last thing I want to say on this particular topic is that the structure of these biofilms, when we manipulate them by driving the biofilms into a greater portions of zDNA, we see much stiffer biofilms. When we drive bacteria biofilms back with reagents or conditions that drive bacteria to bDNA, the biofilms become less stiff. And all this is consistent with our model. So I'm going to shift gears in the last 10 minutes. And I wanted, one of the things that Lauren Bakelitz and I were constantly talking about for a lot of different reasons. We have a whole different story that perhaps I'll get to tell some other time about what we thought was a functional ortholog of the DNABII family in eukaryotes, and actually developed into a completely different story. It turns out that there are proteins that are released from eukaryotic cells that combat biofilms by disrupting the Holliday Junctions. It's a completely different story for another time. But we were very much interested in the host and how the host responds to biofilms. And so one of the big problems with biofilms is that professional phagocytes cannot do anything about a biofilm. They can swallow a bacteria and kill them, but a biofilm is formidable. They cannot phagocytize a biofilm. And so one of the primary weapons of how the host gets rid of biofilms, it creates something called a net, neutrophil extracellular trap. Now, I am just going to say that there are multiple ways you can induce a neutrophil extracellular trap. There could be slow and fast induction, and there are different ways to do it. But ultimately, they have similarities. And the big similarities is these phagocytic neutrophils release their DNA like tentacles from an octopus or a squid. And on those tentacles diffuse modified histones, among other antimicrobial agents. And the DNA acts as a concentrator. And so the DNA acts like tentacles. It surrounds the biofilm, so the biofilm cannot proliferate. And any bacteria that are released get killed by these antimicrobial histones. And so this is, so the host has given up trying to get rid of the biofilm. The host's job is to figure out where the biofilm is and surround it with these nets to stop it from proliferating. And in doing so, basically prevent biofilm infections from becoming chronic and recurrent. So if you recognize the irony that bacterial biofilms use extracellular DNA as a shield, and nets use their extracellular DNA as part of their weaponry, and the two have to interface with each other at some point, I find that irony incredibly powerful. And so the question is, when they do meet, what happens? And so the first thing we wanted to know was, is there any zDNA in a net? And the answer is no, there is no zDNA in a net. And these are nets that are formed by a reagent called PMA, which induces net formation. These are shown right here. But when we add DNABII proteins to the net DNA, you see that zDNA suddenly appears. The B flips to Z. I'm not saying that DNABII proteins are necessary and sufficient to form the zDNA, but they are necessary. And so this was mind-blowing to us that it's possible that DNABII proteins that aren't bound to the eDNA of bacteria could, within the diffusion radius, actually affect the DNA that was coming from the nets. And to prove that the nets were actually being functionally inactivated, we created an assay, a killing assay, to show the ability of PMNs to kill bacteria. So we form a biofilm and we add PMNs. We have data that I have not shown here that show that the PMNs form nets just being in the presence of the bacteria, and they do not kill any bacteria. I'm sorry, this is just the control. When we add the neutrophils, we see about 50% killing, and this is consistent with the bacteria that are being released from the bacteria, or the planktonic bacteria that are not associated with the biofilm. When we add pulmozyme to the biofilm, which is a DNase, the nets no longer can kill the bacteria. But when we also add a DNABII protein, which we know shifts the equilibrium of bDNA to zDNA, the nets no longer can kill. If we use another DNA bending protein that we've shown also is present extracellularly, but does not have anything to do with the structural integrity of the biofilm, the nets do not kill anymore. And so to drive home the story, we looked at 11-day-old biofilms that were created in the middle ear of a chinchilla. And when we look to see where the PMNs are, so down here, this stain recognizes eukaryotic cells. These are the epithelia in the middle ear of the chinchilla. These are all PMNs. You can see how big it is. And this section right here that's black, that is not recognized by the stain, is where the biofilm is. And then we asked, where is the zDNA? And the zDNA is everywhere. It is throughout this entire section. This is 2 millimeters thick, and it's throughout the entire section. We then asked, where's the bDNA? And the bDNA doesn't really start until about here, which is roughly 3 diameters of biofilm away. There's no bDNA at all. All of it's been converted to zDNA after 11 days. We then looked at individual portions, and you can see that right at the biofilm, it's all zDNA. If we move here into this region where we don't see any bDNA, it's still all zDNA. You don't have to go very distal to the biofilm before you start to see bDNA at all. So what we think is happening is that not only is this zDNA structure part of the defense of the biofilm, the DNABII proteins act as an offensive weapon to deactivate the nets. Now, I can tell you that if the histones that are on the bDNA of the nets and get turned into zDNA, the histones get released. And when histones get released, histones are not just antimicrobial towards bacteria, they cause cellular damage. They cause massive inflammation and damage to peripheral, collateral damage, if you will. And so the biofilms, if they are robust enough to release enough DNABII proteins, will in fact cause collateral damage via the nets. And also, because the nets become inactive, allows them to proliferate and cause chronic infection. So we're almost out of time. I'm just going to finish up. This is basically the equilibrium of the eDNA forms. In a native biofilm, they form this kind of lattice structure. When we sequester the free DNABII proteins, the biofilm collapses. When we add cerium, we drive the reaction so that we get more biofilm. We add chloroquine, we get the reverse. And when we add DNABII, we reinforce the structure with making it more, driving it more towards the DNA. So last part, and the part that I often find that people struggle with. So we're going to talk about DNA topology. And so if I could give you all rubber bands, this would probably be useful. I don't know how many of you are experts in DNA topology, but this is going to be a quick background. So a rubber band is very similar to a circular piece of DNA. It has elastic properties associated with it. And when it's relaxed, we refer to that as being untwisted. If you take one end of the rubber band and you hold it taut, constrain it, if you will, and you take the other end of the rubber band and you twist it, you will be adding artificial twist to the rubber band away from its ground state. And if you allow the rubber band to collapse upon itself, it will wrap itself around. Those twists get converted into supercoils. So if you take a rubber band and you do this little experiment, you can do your own DNA topology experiment. So let me run you through 2 more slides before we get to the model. Let's assume that you have there are 2 10.5 base pairs per turn in bDNA. If we have a 210 base pair closed circular piece of DNA that's completely relaxed, there will be 20 complete turns of the DNA. The topology of a closed circular DNA can be represented here by linking number. Twists plus RuvA is supercoils, equals the linking number. When the linking number is equal to the twist, there are no supercoils. If the DNA is undertwisted, then you get negative supercoils, and if DNA is overtwisted, you get positive supercoils. So right now, the DNA is very happy, it's relaxed, and the linking number is equal to the relaxed state, or L0. If I take the DNA and I break the 2 strands, and I underwind it 2 full rotations and ligate it back together, then the twist is at 18, and the linking number is 18. But the linking number is less than L0, and as a consequence, the DNA does not like this because the bases are not stacked properly anymore. The phosphates are too close together, and as a consequence, it wants to get back to a ground state. To do that, it adds 2 supercoils. So by adding by reducing the number of twists by 2, I get 2 supercoils. Likewise, if I were to break the DNA and over twist it twice, I would get 2 positive supercoils. But here is the important point. If I break the DNA and I underwind it twice, and I get this state where I would normally get 2 supercoils, this is C right here, I can transform this by creating this structure here, by melting the DNA, like pulling the strands apart to rewind the DNA, if you will. And so you will find that in biology, supercoiled DNA often has biological consequences that are necessary for the cell. For example, you could argue right here, this would be great to have supercoiled DNA because it reduces the energy for melting DNA for transcription or DNA replication. Likewise, I can transform this negatively supercoiled DNA into this structure, and you can see how proteins that are known to wrap DNA around them would prefer supercoiled DNA over linear DNA because they don't have to do, the DNA does not have to go through this transformation. It happens because of the free energy that's available by negatively supercoiled DNA. So how do we reconcile all of this in conversion of B to Z? So one of the things that's been known for decades is that, and actually there was a very recent paper in nucleic acid research, I strongly recommend that you take a peek at it, where supercoiling, the more negative supercoils that you have in DNA, the less energy that is required. In other words, the greater ability to drive B into zDNA, regardless of DNA sequence. This is a very profound effect. And so one of the questions that we had is, how is it that the bDNA gets transformed into zDNA, but once the zDNA is formed, it can't go back again? And so we came up with the following model. And there are caveats to this. This is just the beginning of the story. I don't have the answers to all the questions, like how do the Holliday Junctions form? We can discuss that. But if you were to have 2 Holliday Junctions form, you would find that the DNABII proteins would migrate between the bDNA that's present, as well as the Holliday Junctions. They would be in equilibrium, go back and forth, back and forth. Once a polygon is formed, and it could be as simple as a triangle, it could be a square, it could be a pentagon, it doesn't really matter. But once this is closed, then that transformation of moving the protein from the bDNA to the Holliday junction, because the DNABII proteins wrap the DNA around themselves, when they leave a closed structure, they supercoil DNA. So if you wanted to supercoil DNA, how would you do it? And so one of the ways is to take linear DNA, add chloroquine, can add DNABII proteins, and then you ligate it together. And you remove the chloroquine, which underwinds the DNA, or the DNABII proteins, and the DNA would lose that wrapping, that supercoiling, and as a consequence would undertwist, and then you get the rive, and you get supercoiling again. So once this is a closed structure and the DNABII proteins are bound here, they're supercoiled, and at this point, once they convert, that reduces the energy of converting B to Z. Once there's zDNA, the DNABII proteins can't go back again, because DNABII proteins are bDNA binding proteins and they can't go back. So we think that this structure, once it forms, so long as the DNABII proteins are bound to the Holliday junctions, that this structure is stable, and all our evidence supports that. And so I can tell you that the first set of experiments that we're trying to do is to create a in vitro model where we create probably triangles, a DNA origami, and show that we can recapitulate these structures with DNABII proteins. Now, having said this, someone has already done the inverse experiment, the reciprocal experiment. They've taken DNA origami and they've taken sequences that in high salt would normally flip into zDNA. And once they're locked into the DNA origami, they will not flip into zDNA anymore. So they've done the reciprocal experiment. We want to do the experiment where we actually show that if we make a DNA origami with DNABII proteins to stabilize it, will we in fact lock in zDNA and not be able to revert it back to B? And this can be done several different ways, but probably with immunofluorescence to start with. So I just want to acknowledge all of my collaborators from the Bakelitz lab, from my lab. Paul Studley was a collaborator, and Santiago Partida Sanchez, his lab helped with all the NET experiments. He's an immunologist, and our funding is from the National Institutes of Health. And I'm happy to take questions.
Thank you very much, Stephen. I really enjoyed your talk.